Hello Brain Shakers, welcome to the Brain Shakers Academy. I am your host today, Brave Alice, and in today's session we are going to be looking at the different cell organelles. We have done a couple of videos, and one of the videos that we have just done is a video on the cell itself, what a cell is, the different types of the cells, and we did label those parts and made mention that the cell within its cytoplasm contains those various organelles, and we are going to be looking at those various organelles in today's session. And now let's quickly dive into today's session and look at those different organelles. So we are going to be starting with the nucleus. I did make mention that the plasma membrane is the lining here that you see on the outer aspect, and we are going to look at it in a separate video alone because it is a complex phospholipid bilayer that requires an in-depth understanding. But let's quickly start with the nucleus, which now is the control center of all the activities that are happening within the cell. So this nucleus here, we did label this as our nucleus. And when we look at the nucleus itself, we're going to have an inner part, and then outside that inner part you will have another lining. So it is a membrane-bound organelle. Now, because it is a membrane-bound organelle, inside you're going to find what we are going to call the nucleolus, and then you will have a nuclear membrane. So this is a nuclear membrane. Okay, so the nuclear membrane in most cases is going to have a number of pores, okay, that allow certain elements. It's selectively permeable as well, allows certain elements to gain access into the cell and certain elements to gain access out of the nucleus itself. So meaning that everything that is produced within the nucleus will then have access to get out of the nucleus. And an example is the ribosomal ribonucleic acid, which is the R, which is a small r, RNA, which is going to be needed later on into the cytoplasm to then cause a translation of the messenger ribonucleic acid. So within the nucleus, you have the nucleolus. Its primary function is to basically bring about the simulation of this ribosomal ribonucleic acid, which will then later be needed into the cytoplasm. And within the nucleus here, you have this region. Now, this region is a region for chromatin. This is where you find mass of DNA, and the mass of DNA that you're going to find in there takes up a form of chromatin. So when it takes up the form of chromatin, this is when the cell is in its growth stage or in its resting stage. It will take up the form of chromatin. It may take up what we call a heterochromatin, or maybe what we call euchromatin. Okay, this is when the cell is in its development stage. But when the cell gets into the active cell division stage, then the DNA material that is within the nucleus is going to be expressed as the chromosomes, getting ready for then the separation or the process or the entire process of cell division. And I have looked at cell division where I look at in-depth mitosis and meiosis as well. And if you have not yet seen that video, you can find that on the YouTube channel, which is the Brain Shakers Academy. So that is your nucleus. It does control all the other activities that are going to be happening within the cell, and that is why it is called the control center of that particular cell. Now, if you notice that from the nucleus, you can also see that you have membrane bound folded channels there, and that is what we refer to as the rough endoplasmic reticulum. Now, what is the rough endoplasmic reticulum? So the RER, or rough endoplasmic reticulum, is an important structure that has ribosomes on top of it, and that is why you see those dotted spots around those membrane channels, because it is responsible for the synthesis of protein. So one of its functions is protein synthesis. Okay, so there is protein synthesis, whilst in the smooth endoplasmic reticulum we will not have those ribosomes. So the rough endoplasmic reticulum is going to have ribosomes onto it, and those are the ribosomes that are then responsible for the conversion of the mRNA into proteins, or the translation of those mRNA into proteins. And so it is responsible for protein synthesis. It will be used also as storage, and then it will be used as transport media or channels, and then plays a key role as well in detoxification. So it plays a key role in the detoxification of drugs, alcohol, and other toxins that will then enter into the cell. So similar functionality also happens with the smooth endoplasmic reticulum. So you have the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum will have all these other three functions, meaning that it is going to play a key role as well in storage. It will play a key role in transport. It will play a key role as well in the detoxification of toxins, of drugs and alcohol as well. But in addition to those functions, the smooth endoplasmic reticulum, which is also a membrane-bound organelle, another function that it is going to do is that it is going to be in the forefront of the formation of lipids. It will be in the formation also of phospholipids. Okay, so these phospholipids, then they come on and be a part of the plasma membrane, as we are going to see when we look at the plasma membrane. And then you will have also steroid synthesis, meaning that all the other steroid hormones, they will have their precursors as a result of the smooth endoplasmic reticulum. And this same smooth endoplasmic reticulum creates an environment also for carbohydrate metabolism. So meaning that all that metabolism for those polysaccharides and other disaccharides or monosaccharides, all those carbohydrates will happen within the smooth endoplasmic reticulum. So those are the two different endoplasmic reticulum. And the other function that we can actually relate to the smooth endoplasmic reticulum is when you get to the musculoskeletal system, especially if you're looking at muscular contractility. The smooth endoplasmic reticulum in the muscles now is going to be called as the sarcoplasmic reticulum. So that sarcoplasmic reticulum is going to be responsible for the sequestration of calcium cations. Now, you need calcium for a muscle to obviously go ahead and contract, and that will be the response now of that reticulum within the muscle cells. Now, having looked at the two different reticulums or endoplasmic reticulums, we can go ahead and look at another membrane-bound organelle, which is the Golgi apparatus here, or it is also called the Golgi complex. It is also called the Golgi body, and it is actually named after an Italian scientist. Now, the role of the Golgi apparatus is that it is storage and transport. So it plays a key role of storage and also the aspect of transportation. Why? Because it is membrane bound and it creates all these large surface areas so that it can then carry a number of elements that have been produced by the cell, and then it will have the budding vesicles from the Golgi apparatus. So it is formed of what is known as a cis and a transverse, and then that is where obviously you have these elements now budding off from that Golgi apparatus in readiness for them to be utilized. They are either going to be gotten rid as exocytosis, or they are going to then be utilized by the cell itself. Now, you also have what is known as peroxisomes. These peroxisomes are an important element within the cell as they play key role. Peroxisomes, as they play key role in the process of oxidation. So they play key role in the process of oxidation, and they also house the proteases, which are enzymes that can then be stored within them for utilization whenever the cell needs those elements to come into play. You also have another important element, which are lysosomes. Now, when you look at the lysosomes, we can also look at the vesicles together. Okay, lysosomes and vesicles. So the vesicles, there are three main types of vesicles. The number one vesicle that we can talk about, we can talk of exocytotic vesicles. Then we can talk of secretory vesicles. Then we can talk of lysosomal vesicles. Okay, so these are the three main vesicles that we are going to see. This one is what we would call a secretory vesicle, and then you would have obviously vesicles like coming from the Golgi apparatus that can also be maybe exocytotic vesicles, and then from the lysosomes you have lysosomal vesicles. Now, what is an exocytotic vesicle? So exo is out of the cell. Cyto is a cell, so meaning that out of the cell. So all the produce that is going to come out of this cell that has to be transported to the other elements or to other cells outside this cell which is actually producing it, then are going to leave the cell through vesicles that we're going to call the exocytotic vesicles. Now, the cell can produce elements that are capable of changing the way it functions, and also produces elements that need to then be transported to other target areas and target organs to go and cause an effect on those target organs. Now, if the cell does not want those elements that it produces to cause any conformation or change the chemical environment of its internal milieu, what it is going to do is it will package those products into vesicles, and that is why a cell has these vesicles. So when it produces those exocytotic vesicles, a classical example that I can give here is when you have a cell such as a B cell. So you have obviously T lymphocytes and you have a B lymphocytes as well. So if you have a B cell, we know that mainly the antibodies are coming from B cells. Now, if this is a B cell and it wants to produce those antibodies, what is going to happen is that it does not require the antibodies, but it has been stimulated or has been given information that these antibodies that you do produce are needed in a place called place X, meaning that it will have to produce those antibodies, package them into an exocytotic vesicle, and then have them out of the cell until they reach the target area, which is a place X for them to actually work. Then that will be a process of exocytotic or the use of exocytotic vessels. Then when you have secretory vessels, secretory vesicles also are working in having the produce that has been produced by this cell leaving the cell. But in most cases, this one is going to be under stimulation. A classic example that I can give is when you have a neuromuscular junction. So what happens is that you have the first neuron there, then it will have its telodendria, then you have the postsynaptic neuron here. So this first neuron that is coming there will have been stimulated by an action potential, and once it is stimulated by the action potential, what is going to happen here for this other neuron to actually be excited, there will be need for the release of a neurotransmitter here. So the neurotransmitters are usually packaged into vesicles that will then move towards the end of or towards the telodendria there, so that these, when they bud with the plasma membrane, then they'll be able to release their acetylcholine straight into the synaptic cleft and then be able to excite the postsynaptic neuron. So this is a classical example of how the secretory vesicles are actually going to be released or come into play. Then you have the lysosomal way or the lysosomal vesicles. These ones, what normally happens is if this cell has been invaded by any pyrogens, what is then going to happen is that the pyrogens or organisms that have invaded it, or any organelle that has now expired or has reached its lifespan and it has to be gotten rid of, there will be a vesicle that will then form around that particular organelle or virus or bacteria or fungi, whatever has invaded the cell, and then this vesicle is going to get attached to a lysosome. so that the lysosome, which contains those hydrolases, enzymes, proteases, is going to bring in and release its elements into that vesicle so that no any other organelle within the cytoplasm is affected, and then it would degrade this particular protease that is contained within the vesicle. So that is how you are going to have those vesicles coming into play, and the lysosomes as well coming into play, mainly for purposes of defense mechanism. And then now you have the mitochondria. So the mitochondria is referred to as the powerhouse. So the mitochondria is the powerhouse. Why? Because it has two main membranes, and within the two main membranes you have a space in between, which is referred to as an intermembrane space, which is the space in between. So you have an outer membrane, an inner membrane, and then you have an intermembrane space, and this space in between here is what is referred to as the matrix. You have a little fold. From time to time, you have all these folds within the mitochondria that are going to be called cristae, and basically creating more surface area. And it is the only organelle that contains its own DNA material, and so it is capable of replicating itself. And you're going to find these mitochondria mostly in abundant amounts in cells that require more ATP or more energy sources. ATP is just adenosine triphosphate. So when you get to the cardiomyocytes, which are the muscles, the cells that are found within the heart, you find that these mitochondria are actually in abundant amounts because the heart needs a lot of energy for it to continuously pump and maintain that particular rhythm. So within these marked X's is where the Krebs cycle or the citric acid cycle also does happen. So it is a central portion for the generation of the adenosine triphosphate. And I look at the citric acid cycle under a component of biochemistry as well in a separate video. So make sure you're subscribed to the YouTube channel so that you don't miss any of that. So that is the mitochondria. It is basically for the generation of adenosine triphosphate, which is the energy that is required for the cells to actually function. You have another important element here, which is the centrioles. The centrioles come into play when the cell is about to divide, meaning that they're going to move to the poles of the cell, one this side and the other one on the other side, and then they will have those microtubules coming around them, then forming the mitotic spindle and allowing the chromosomes to then align, or the chromatids to align themselves on the metaphase, and then they allow the process of cell division. It means that this one parent cell has the capacity to then divide and form two daughter cells that are going to contain similar characteristics to the parent cell. The vacuole here is just utilized basically for storage of cell elements or products of the cells. Now, you have free ribosomes. Most of the ribosomes that are there are attached to the rough endoplasmic reticulum, and some of the proteins that are produced by the rough endoplasmic reticulum are proteins that are then going to be expelled out of the cell. But the free ribosomes that are there are the ones that are then producing the proteins or the building blocks for the cell itself. Now, you also have these microtubules that are very important. The microtubules, like we have made mention, that they play a key role in the process of cell division. Now, just outside the anatomical aspect, just for clinical application, the microtubules are formed out of what we call dimers, called the alpha and the beta tubulin. Now, the alpha and beta tubulin, when they are put in long arrays, then they become axonemes. So they will form, and then they will help the cell maintain its integrity. They will help the cell as well in locomotion. And when you are giving drugs such as alkaloids, as drugs, for instance, such as vincristine, okay, when you are treating neoplasms or cancers and you give drugs such as vincristine, drugs such as vincristine have the capacity to come and block the tubulin dimers, meaning that the cell, because you have these microtubules that then allow the cell to have all that mitotic spindle, and you have the chromatids align on the metaphase. When you give this vincristine, what it is going to come and do in that cancer process is that it will come and block the formation of those mitotic spindles and arrest the cell in metaphase, meaning that there is not going to be any separation of these cells. So as a result, the cell is then not going to replicate and form new daughter cells, and then the process of metastasis then is kind of delayed or actually blocked or completely stopped. That is how certain drugs like alkaloids, like vincristine, will be used. It is because of their capacity to come and block the formation of those alpha and beta tubulin dimers within the microtubules. So those are the organelles, and basically that is what forms the cell. Those are the organelles of the cell. And if you found this particular video helpful in understanding the different types of organelles and what they do in a cell, don't hesitate to drop your comments in the comment section. Give it thumbs up, share the video as much as possible, and don't hesitate as well to go ahead to the YouTube channel, subscribe, hit the notification button so you don't miss any of the amazing content. And as always, thank you so much for watching, and I will see you in the next one.